So okay, once again I welcome you all. Guys, any problem there? Yeah, we can start the program. You can't? Yeah, then come half of the can come this side because camera is this side, so I cannot come there. You can come this side. Anyway, you can see the slides there, no? You can come this side. Half of the people can stand. Yeah. What's his name? Few of them can come this side. Move forward. Yeah. Yeah. You can still see from here. Don't worry. Come, Monica and what's his name, brother? Yeah. Come this side. Anyway, experiment part will be done here only. Just I'm showing the slides. So today, what is the work plan? Okay. So majorly, we are trying to transform the nucleus to the nucleus. Morning we talked about chloroplast transformation, no? But we are not doing exactly the chloroplast transformation. We are doing nuclear transformation. But the method will be almost similar. The instrument which you use will vary. Here we are going to use electroporator. Since due to the availability, we are using electroporator. Majorly, I have planned everything like cloning, transformation. Cloning will be a big step actually. And in order, it will be a big process. So I will be telling you theoretically how to be done. Because doing experimentally, it's nothing. Because it's like a digestion, you are going to add. Everything going to happen invisibly. So by doing, you are not going to see much. But I will be explaining you how to be done. That part I will be taking care. But experimental, you will be starting transformation. You need to imagine a way that plasmid which we are going to give contains your gene of interest. From then onwards, you will be going ahead. You will be transferring the vector, transformed vector, into the DH5 alpha. Why we are doing that? Yeah, competent cells. DH5 alpha is a competent cells. What is the importance of DH5 alpha? Why we are using DH5 alpha cells? More copies? No, not exactly more copies alone. There is one important point, genetic, I can say advantage. No, not high replication. What is the difference between wild type E. coli and DH5 alpha E. coli? No. Multiple cloning sites? No. It's multiple cloning sites comes in the vector. I'm talking about the organism. The E. coli, which we use is a DH5 alpha that has some mutations. So what is the feature of DH5 alpha? Why specifically we use this DH5 alpha for the transformation? That's my question. So I will be giving you the answer. DH5 alpha contain mutation in the recombination. It cannot recombine. So whatever the gene you are giving, it will not integrate into the host. So they will replicate. So that whenever you want to isolate, you can isolate them. And whenever you want to store them, you can store them as a extracellular. They won't integrate into the... Yeah. Sorry, I'm not...
Here you guys are using
DH5 alpha cells. So mostly any lab, generally they use DH5 alpha cells for storing, for copying, making copies, everything. Easy for growth and easy for... Top 10 cells also. Fine, that is also. But this is one of the primary and easiest to grow. Top 10 is a eukaryotic or prokaryotic? Prokaryotic one. Then I think there is no difference. There are some vectors, some organisms, they store it in the eukaryotic. Depends upon the gene of interest. But here, if your idea is to store and to recover high copy plasmids, DH5 alpha works better. You're asking something? Yeah, because DH5 alpha cells, they have a mutation in the recombinase. As a result, they cannot be able to recombine. Whatever the plasmids you are giving, they will not integrate into the chromosomes, nuclear chromosomes. So that's the reason, because whenever you want to isolate the plasmid, you should isolate. If it integrates into the nuclear genome, you cannot be able to isolate, right? So that's the reason we keep DH5 alpha. So you will transform your plasmids, whatever the plasmids, we use heat shock method. You will be going to do the heat shock method. So you will be transferring whatever the vector, it doesn't matter. It will take up your vector and it stores. And whenever you want, you can put it in the LB media, grow it overnight. You can isolate the plasmids. So it acts as a storage as well as for multiplying the copies. Two purpose DH5 alpha is used. Anything? Fine. Okay. Then finally we will be going for the transformation using the electroporators. We use the electroporator today for transformation. So simple. Any doubt in the outline today? So first I will be giving you the theoretical explanation how the cloning will be done. So what is cloning? I think she might be knowing. Okay, I will come to you later. Let's ask the students. Last answer will be yours. From this side, I forgot your names actually. What's your name? Shamila. What's your name? Asmita. Yours? Pranati. Okay. What's yours? Subhashri. Yours? Dashrath. Okay. Okay. Any of you? Come on. Let's say who can say the better clear definition.
Means, simply making many copies of the same organism.
No, cloning is not making copies.
Other foreign.
Oh, yeah. Make it very clear. Yeah, cloning is nothing but you are inserting whatever the fragment, whatever the gene of interest into the vector. That is called cloning. And it involves two steps. One is digestion, digesting the plasmid, because plasmid has, morning I was telling you, different parts, right? In that multiple cloning site I said you, where you will be inserting your gene of interest. That multiple cloning site, for example, let's take this is the vector which we are going to use today. That is PSL 18. It is well standardized for the Chlamydomonas for expression. It has PSA D promoter and multiple cloning sites are here. Okay, and this is the promoter, PSA D promoter, photosystem protosystem 2 promoter, followed by this is the multiple cloning site, and it has many restriction sites. You can see these are all restriction sites. Particularly this region, we can use whatever the restriction enzyme available to you and compatible with them, we can use them. So this is the vector. Enzyme is not present in the gene of interest. Why the second point is important? Why the restriction site should not be present in the genome?
It will cause the gene of interest and we do not get our
Yeah, exactly. So the restriction enzyme you are keeping should be unique. It should not be present in the target gene which you are going to clone. So how you will identify what are the restriction enzymes present in your target gene or not? Is there any tool? So there are many tools out there. I use two tools. One is SnapGene. SnapGene, once you give a sequence, it will say what are all the restriction site available. As well as if you go for NEB cutters, if you give your sequence, they will be telling you list different 1 cutter, 2 cutter, 3 cutter, and there's an option called 0 cutter. So I will be showing you 1 second. So this is NEB cutter. This is one of the software, I can say. So they changed it. It's latest version. I think Wi-Fi is a bit slow. Yeah. Let's take, I'm randomly taking some sequence and I'm pasting in the 0 cutter. Okay, this is the dialog box. I'm pasting my sequence. Now you can see there, same thing I've done. Then I'll be submitting. So you'll be getting the list of restriction sites, right? So here the one which is selected is one cutter. So if you go for enzyme list, these are all these whatever they mentioned will have single cuts in your it shows where it is also, but you need zero cutters, right? You select zero cutters. So these are all whatever listed, right? These enzymes were not present in the sequence which I have given. So you can select any one from this, but still you need to cross check whether that enzymes are present in the vector. So in this way you should select the restriction site, and you should design your primer in a way that it should amplify along with this restriction enzymes. Okay, now I'll be back there. So in this way you need to design your vector, and you need to amplify your gene of interest with the 5' end should be having the restriction sites. So then you will be amplifying and you will be digesting, like here you can see, no, we will be digesting both the plasmid as well as the gene of interest. So here also sticky ends will give better results for ligation. There are two types, no, blend end and sticky ends. So people always prefer for going for sticky ends, but in unfortunate situation you are not getting any compatible restriction enzymes, then finally you should go for the blend ends, but last option should be the blend ends. And then you will be going for the ligation. I will be telling what is ligation. Okay, I think here I haven't kept the slide. So okay, no problem. Ligation is nothing but you will be incubating the digested two products are there, no? So how you confirm it is digested or not digested, the plasmids? So no, you are digesting with the enzymes, but how sure that they are digested? Yeah, you should run on the gel, right? So you will be running on the gel. So generally undigested plasmids will have how many bands theoretically?
Undigested. Single.
Yes, sir, telling single band. Anyone? Undigested plasmids should have how many? One. Wrong. Majority is wrong. Undigested plasmid will have 3 bands theoretically.
Like they shape.
They actually supercoil. 3 bands. You remember? There will be 3 bands. So generally, people don't try to run in the undigested plasmids because they will get 3. They will confuse. So you know plasmids are circular. So when you are going to run, there will be 3 possibilities. One is they supercoil and they become very short, compact. So they will be the least lower bottom, one band will come, and there will be a middle. That will be a niked one. Niked one means unfortunately one cut happen, they will become linearized. So linearized will be in the middle, and the top one will be the complete DNA what you expect. So when you run a normal without undigested plasmid, you will get 3 theoretically, but generally we will get 2 because the linearized one will be rare to get, but the complete open plasmid and the supercoiled plasmid you can see always. But when you digest now, how many bands you expect? If you digested, the digestion is complete, you should get single bands. If undigestion is there, then there will be 2 or 3 multiple copies. Then you need to identify which one is the right one. For that you should keep a control. For example, here in this case you are digesting with 2 restriction enzymes, right? But you got let's take, instead of single bands you got 2 bands. So how you analyze this results? What can you say?
So can you repeat the question?
You want to digest the same, like you are digesting the plasmid with 2 restriction enzymes. You expect a single band, but you got 2 bands. What will be, how you analyze this result? What you say?
Digested.
Partially. It would have been partially digested, or one enzyme would have worked, another enzyme would have not worked. So you need to keep negative controls. Like individual restriction enzyme, you need to digest and check whether both the enzyme can able to give digestion properly. And sometimes incubation time, if you increase the incubation time also, the results will be better. So you need to try different methods to find out which works better for you.
Yes, control means the DNA which was well digested.
Control means without digestion.
Yeah.
Control means without digestion. You will be loading then single enzyme. That will be a first treatment. Then enzyme 2. Then enzyme 3. Means enzyme 3 means dual combination. If both the single gave a single fragment, that means both the enzyme work properly. Then more likely the third one also should work. If either of these two enzymes are not working, then you can predict that which enzymes are not working. Okay. Okay. Any doubts here? Yeah, that's what I was telling. No, you need to go for the NEB cutter. It depends. It's not the particular enzyme you're going to use for all the experiment. Yeah, in this, the gene, right now we are not going to use any restriction enzyme. I'm just explaining how to do it. So if, for example, if I have a gene, let's take hypothetical condition. I am checking in the NEB cutter. Right now I was checking, no, in NEB0 cutter I got few restriction enzyme, and I will be cross checking. Let's take, I got a NEB0 cutter eco R1, and I will check here eco R1 is there. Okay, then I can put eco R1 in my gene of interest. Similarly, I will look for another restriction enzyme for the other side. So like this, I will be taking two enzyme and I will be amplifying my target of interest with those restriction enzyme. Okay. Then after digestion, you confirmed with the gel, you need to go for ligation. You need to gel elute, purify. Then you need to use the T4 DNA ligase. So what is the purpose or point of ligase? What does it does? Phosphodiester bonds, right? Means the DNA will come in close proximity because of the random molecular movement. But the sealing has to be done by diester bonds. So that will be done by the help of the T4 DNA ligase. And generally we keep overnight. We keep it at 18 degrees Celsius overnight. And the next day morning, what we will be doing, and let's take 1 microgram of DNA initially for digestion. And you will be ligating all of them, right? And you are going to transfer the next day all of them. But in the 1000, let's say 1000 microgram, hardly 1 or 2 plasmids would have ligated. It's highly probability based, right? Not everything will be ligated. So we need to find out which one has ligated, which is not ligated. So for that only we are going to transform into the DH5 alpha cells. So what is the advantage of transformation into the DH5 alpha?
You can make out express.
Yeah, if they are sealed completely only, they can able to express. If they are not sealed, they cannot able to express. So how we are going to select? We need to check any antibiotic resistance. Here in this case, ampicillin resistance is present, right? Oh. No, here ampicillin resistance is there, LAG-Z is not there. You can use LAG-Z also there, yeah. LAG promoter is there, no? We can use two things. Generally, if you want to use LAG-Z also, you can use to check whether insertion is there or not. So ampicillin resistance, once you transform into the DH5 alpha, you will be growing them in the ampicillin, LB ampicillin plate. So what if the colony appears the next day? Most likely they should be having the sealed plasmids with gene of interest, but still you need to confirm. How you confirm the plasmid has your gene of interest or not? Gel? Gel is not the first step. No, selection we are growing in the plate. Ampicillin is not a strong antibiotic. Sometimes artifacts also can grow. But how you are sure that your gene of interest is present in the plasmid or not? Yeah. How you confirm? You need to do some confirmation, no? Tracking?
First we'll check the OD.
Okay.
And then we will go for
What?
You can isolate.
Yeah. You can do isolation, but that is a little tedious job. Let's take you have 100 colonies in your plate. Your transformation is very good. 100 colonies in this. In this 100 colonies, you want to select which is having positive colonies. How you will do? You will isolate 100. 100 is tedious. You want to do it. You can do it, but it's a very tedious process, right? How? How? That's what I'm asking. Screening only how? No. You will do colony PCR. Simple. You will pick a colony and do a PCR with the G primers, same primers of gene of interest. And if you are going for any expression study, it is very important to check the sequence. You need to send it for a sequence to confirm whether the open reading frames are fine. Sometimes mutations may happen. because you are PCR amplifying, right? So you should be very careful. Only if you confirm the gene which you have present is exactly the same, then you can proceed for the chlamydomonas transformation. So now we are going to start from, now you guys remember that you imagine that we have cloned. So we are isolated the plasmid. So we are going for DH5 alpha transformation. From now onwards, we'll be moving ahead. First step is you are going to do is transformation into the E. coli. Okay. So this is like transformation, isolation of the plasmid from the E. coli. This is, I already explained you. This step only we are going to do now. First the plasmids, we already have the PSL18 plasmids. So now the protocol book I given to you, no? Do you have that now? That day each group we are having, no? This is one I made it.
Monica, Monica.
Yeah, we can start.
Can we use it for the cloning method only for
Yeah, of course. Means for inserting your gene of interest, right?
TA cloning?
Yeah, of course. Anything is fine. This is very primitive. TA cloning you can use, gypsum you can use.
Someone can get ice first. I get the ice tray.
Yeah, one ice box is enough. Give one. Yeah, one can go and collect the ice. No, vector is not DH5 alpha, sir. Vector is a PSL18.
Who knows where the ice machine
It's
is?
a cell, E. coli. In that only we are going to transform. Okay, then bring the
She's
plasmid. Okay, okay, once she brings.
We're going to take out the
Just follow this, what other things required. PSL18. It's well used for overexpression.
LBNO is with you.
Sorry? Yeah, any vector you can use. Method is same. I'm just showing you with one vector. You can use it for the same for any vectors.
Sir,
And one more thing I forgot to mention. The ligation has different combinations. Vector to fragment, there is a molar ratio. How you will calculate? It's not like 1:1 ratio you will take. It will, based on the molecular weight, you need to identify what is the best matching concentration you will take. So for that, there is a tools if you really want to use. There is NEB cloner. So like NEB zero cutter, NEB cloner is there. So if you give your plasmid size base pairs, like PSL18 is 6,000 bases, and your gene of interest base also like 1,000, 2,000 you mentioned, and finally how much concentration of DNA fragment you are giving, 200, 300, then it will suggest different concentration, which one, 10:1, 3:1. So you can, it's like a random. You always I always suggest you to take more than 2 ratios, because it's completely a random process. It's nothing like 1:1 works. Whenever you're going for ligation, keep 2-3 different combinations.
How through the colony PCR?
Yeah, colony PCR is that day, first day we are doing some colony PCR in algae, right?
Like how PCR helps to identify?
Yeah, that's what. What is the transformation means? Digestion and ligation, what we are trying to do, we are trying to insert our gene of interest into the vector, right? So our aim is the plasmid should have our gene of interest. So that's what, using PCR, we are trying to amplify again the gene of interest from the plasmids. If it can able to amplify, that means gene is present inside, right? So that's what we are trying to. What happened? Why you are changing? What's your name? You please come this side. What do you want?
Got pressed when I was ready.
Yeah, otherwise, if you want to sit, you can sit this side and write it. Laptop. Fine.
PhD data.
Yeah. Okay, any other doubts you can ask me now. We'll just wait for ice. Once ice is ready, we will go. You do agarabacter transformation.
For the dissertation,
Okay.
it was like a new plan.
Okay.
I was trying to standardize.
But agrobacterium for higher plants, no? Which organism?
Agmella oileraceae, Asteraceae family.
Okay. So you're
Common
successful.
plant.
You're successful in that.
Like I was getting it transformation, but it was what is it? Like we do gas expressions. P can be a vector.
Okay.
It has gas.
Uh-huh.
So the spots were like not uniform.
Okay.
I'm getting like spots. In case she will be doing the rest.
So it's like a part of your work, or it's
Like she added my things with
oh. Oh, you already have a publication. Which journal?
like plant culture. Plants that PCTOC.
Please. Any of you has a publication? You. He's from a big lab.
Yes,
Where green biochem. What is the journal? What journal?
chemical engineering.
Okay.
Chapter in my one paper, it's very small journal.
Mm-hmm.
Environmental biotechnology.
Yeah, got it. Keep it here.
This is the vector.
Okay, I'll bring the DH. Okay, I'll bring the DH. Excuse me.
It is now.
Take that, it'll keep it nice. Yeah, one is enough. Wow. This is having what is the concentration, Shreya?
130 nanogram per microliter.
200. Okay, 200 nanogram. Pipettes 10.
This is 1 ml. 1 ml is there.
Okay. Need a gloves. Can you give me a gloves? MM. So who wants to do? One person will show anyway. So who wants to be doing that? You want to do? Who haven't done it till now?
Me,
You haven't
never,
done?
but I want to do. I never done it before.
So who want to give up among you?
No,
Hmm. Okay. You want to do? Okay, let's first step is we have the protocol, no? Just I brought the competent cells. This is the competent cells. Competent cells preparation is you can get it from the protocol from the online. So there is nothing. You need to treat them with the calcium chloride. You need to rupture the cell wall. So it's a very simple protocol. Anywhere you can find it. Once you make the competent cells, you store in the minus 80 degree. Whenever you want to transform, you will keep it in the ice. Allow it to thaw for one minute. And meanwhile, you should transfer your plasmid of interest. The plasmid which we have is PSL 18. We already isolated. It has some 200 nanogram concentration. So first, as it is mentioned, we have switched on the water bath. This is the water bath for heat shock. So 42 degrees Celsius, we need to heat shock. So first we need to take around 5 microliter of the plasmids. 5 into 2 means 1000 microgram we are trying, but that is too high. Maybe let's take 3 microliter, you transform. 3 microliter of the plasmids, you transform to the competent cells and then leave it for a while, 1 or 2 minutes. Then we need to go for heat shock. So heat shock is very specific time, 90 seconds I mentioned, no? 90 means exactly 90. If you keep for even little like 10-20 seconds extra, it will affect the cells. The efficiency of the transformation will get affected. So make sure once before going, you set the alarm. 90 seconds, time of keeping and taking out should be exactly 90 seconds. And then chill on the ice. Immediately you should chill it on the ice. And then you will be adding whatever the transform cell to the 1 ml of LB media. We will grow them in the 1 ml LB media. Why we are growing for 1 ml? LB media we are growing means immediately after the heat shock we are transferring to the LB media, and we are allowing them to grow for 1 hour.
We are making it to allow the insertion of the form.
No, that is done by heat shock. We are allowing to recover. The cell wall should be formed. No, means the entire genetic material will penetrate during the heat shock process. So once after heat shock, we are trying to recover. No, they should recover. The cell wall should be formed. So this 1 hour will be enough because doubling time of E. coli is 20 minutes, right? So that's what in next generation they try to form their cell wall. That's the concept. So then you read it. Next protocol, what is it? I mentioned 1 hour LB media, 37 degrees Celsius. Yeah.
After 1 hour centrifuge cells, 6,000 RPM at RT for 5
Okay.
minutes.
We need to remove the supernatant and we need to pellet down the cells. After that,
Resuspend the cells with 100 microliters of LB media
fresh LB media. Again, we are resuspending the entire cells in fresh LB media.
and spread it on the LB plate.
LB plate with ampicillin. So that step that has an ampicillin, that's a selection marker.
Maintain the positive as well as the negative.
That you can do it. And then finally,
Incubate the plate at 37 degrees centigrade
overnight. Okay.
Cells
So we'll be splitting today and we'll be keeping it overnight. That step we'll be doing. And again, the same plasmid we'll be showing you the electroporation, how it has to be done. Two methods. One is transformation we are teaching, another one is electroporation, algal transformation. In other way, we can say.
Here we are not like getting enough interest with vector.
That's what I said. Today we are not demonstrating that. We are directly going into the transformation step. Yeah.
Already prepared vector into
Yeah.
the areas of
Yeah. Yeah. That's it.
Sir,
Fine.
here we are using 1 ml LP dose and here we are using 100 microliters.
Yeah.
Why that difference?
We are going to spread it on the plate. We should not have more media. No, we should spread entire cells in a single plate. That's a concept.
Is there any alternative for polymerization instead of polymerization? There is no other method.
That's the simplest method. That's why we are using.
So
Otherwise, that should be the most simple media method. Otherwise, sequencing you should do. That is more complicated. Yeah, don't keep it outside too much time.
so is there any
So you are going to do now. Wear the gloves.
so is there any specific ratio that how much of the vector should be transferred to how much of competent cell?
There is not for example, you can take 200 to 300 nanogram. That range.
Of vector.
If you take more vector, you will get more colonies, a lawn. So people don't like to have a lawn. So you need an individual colonies you require, right? So something lesser than 500 nanogram is more ideal for 100 microliter of competent cells.
500 nanogram for 100 micro.
Less than 500 nanogram for 100 microliter of competent cells. Okay. So now take pipette and set it to 3 microliter. Slow. Which side you know to turn? No? Set it to 3 microliter. 10 microliter tip box. 10 microliter tip box.
The thing is,
Yeah. Tip boxes. Shell today, no sir. No need actually for today. Yeah, you can use this. So she's going to transfer 3 microliter. She has taken. Fine. 3 microliter of the vector and transform into the competent cells. Whatever. Oh wait. Give that some more water. Give me that tip box. And again, molecular biology, everything. It's like a demo, so we are giving it in a crowd. But when you are doing it in your lab, try to be isolated, sterile, wear gloves, mask, everything. Yeah. So 3 microliter, take it exactly. Yeah. Fine. We can keep one more negative control. Yeah. And keep it in ice for 5 minutes. It should be kept in ice for 5 minutes. Shreya, can you take one more competentials? We will keep one negative control without plasmid. So technically, negative control should not grow in the ampicillin. Okay. Namrata, I took the plate, LB plate. So it means, I think why we are keeping it in ice, they would have been very frozen state now. They should thaw first, and meanwhile the plasmid will get mixed completely inside. So when we go for heat shock method, it's more likely there will be a green in the middle. Okay, I'm coming. It's written at DH by R. Take one. Later. Later. Okay.
In dark or in
No. This is not at all light sensitive. You can do it in light, but isolation required. Sterility is very important for mold bio rather than light. Keep. So let's make this as a positive control. Marker required. This should be negative control. Label it, otherwise you confuse.
So one is with plasmid and competitive cells, and one is without plasmid and competitive cells.
Without plasmid. One treatment is with plasmid, another one without plasmid. That's it. So that you can see the difference.
Same competence.
Competence cells are same. Only thing you need to understand that the plasmid makes them to grow in the ampicillin plates. Sorry. So make sure always this is there. If it goes, then don't confuse, okay? So now we can go for heat shock. Who is setting the timer? 90 seconds. Timer is perfect. 90 seconds. 90 seconds. Means 1 minute 30 seconds. It can.
Stopwatch is there.
Otherwise phone watch you can use.
Stopwatch. Anyone keeps timer for 90 seconds.
This
Yes,
we
yes.
have already done.
Yeah.
Continue keeping.
Yesterday, then.
Did we do this?
What was?
Watch 90 seconds.
1 minute 30 seconds. Okay, you can see. Okay, you can set it for 1 minute 30 seconds, or you can watch till 1 minute 30 seconds. Anyway, it's fine.
Timer ends.
Okay.
1 minute 30 seconds.
Second. That is 90 seconds, right? 60 is for 1 minute, right?
Okay, start.
Yeah. Means immediately you transfer. Yeah. Yeah. Once the moment she kept, you can switch on. Switch on. Other one also. Negative control also. Yeah. Switch on. So what is the next step? You focus on the. Chill them. You need to keep it in the ice. Immediately take it and keep them in ice. Then LB media. We need LB media. You can take the LB media here. Okay. But I need a Falcon, 5 ml Falcon, 5 ml FNO.
I mean, the vector
Two.
will be
Bring 2. No, unused.
just one. Just heating.
Competence cells.
Why are we doing this? No time.
Not no time. It's like yeah. Basically, time is limited, and moreover, even if I do, it requires a complete sterility. It has to be done in the laminar, not in a crowdy atmosphere. Even if you do in the crowdy atmosphere, it doesn't make justice for the work. So that's why I'm teaching you the theoretical part, and I'm directly moving into the
When have you done
last week. And once you can make years bigger before also. Once you store inside, it's not an issue.
13
Lambda.
What is the temperature?
5,
like
47,
44 degrees.
Sorry.
Is this an expression?
For how much?
It's 1 second. It's done. Just transfer it.
1 minute 30 seconds.
Transfer. Keep it in ice. What you are asking?
The expression vector
Yeah, it's expression vector. That's it. Okay. Use a
for reducing.
for us.
fine. Keep it in ice. Yeah. Let them for 5 minutes. Let them cool. Note down the time for 5 minutes.
You can see how my nothing is.
How?
T7, right? T7.
Yeah. That's promoter can vary anything. It's not an issue. Means this is like we purchased this promoter, vector. It's not we made it. We purchased from Clammy Resource Center. So
1 ml.
nothing, any promoter can be used until it's going to be working for you, right?
No.
The main idea for it's like for any overexpression we can use PSA-D is there. It's a strong constitutive promoter, and it has been widely used in many chlamydomonas like research. Earlier papers were there, so that's what we prefer to buy this plasma.
E. coli is 20 minutes now. E. coli.
What happened?
He's asking if 1 hour it will go. So E. coli takes 20 minutes.
Yeah.
40 minutes.
20 minutes. Dividing is 20 minutes. Okay. 5 minutes. Let's keep. After 5 minutes, what you have to do? You need to transfer 1 ml to this.
We brought to the center.
Yeah. You label which is positive, which is negative. You can label it treatment. Another one is negative control.
Like when I did internship, they were doing silencing, RNA silencing. So they have done gateway.
Great. I use gateway for some complicated, like gateway for guide RNA. When you are targeting multiplexing, they used to say multiple segments you need to incorporate. No, that time this method is very difficult. Restriction, digestion. The simultaneous incorporation of multiple fragments, gateway cloning is a bit advanced to understand. But once you establish it, it's easier.
Nitrogen peanut plant.
Okay.
So
That part.
Yeah. Anything?
I am confused. This in between these two.
What is it? A confusion? Tell me.
Like which tube is containing a vector, which tube is containing competent cell, what
Yeah. Only one. Wait. Only one thing we changed. Two tubes we are having. Both the tubes are having competent cells.
okay.
One is we added the plasmid. That will be your actual
Only
sample.
one.
Only one tube we added the plasmid. That is your actual sample. Another one we kept as a negative control. Negative control we didn't add any plasmids.
Negative control means
Negative control means there should not be any growth. Means we are believing that the growth of the cells in the ampicillin plate is completely because of the plasmids.
okay. That's only competent cell only. We have
Yeah.
not added the vector.
Vector is the difference between the negative control.
Vector nahi.
Yeah.
लेकिन मैं
For any experiment, you keep one positive control, one negative control, right? So in this case, negative control is without the plasmid.
okay.
So we are transferring the vial with competent cells and plasmid to heat and ice.
You can keep the both. Both we kept. Means everything should be the same, right? Positive control, sample and the negative control should be treated with everything the same. Only thing it does not have a plasmid. So that you can say the sole difference between them is because of plasmids, right? For example, if you are not treating the negative control to temperature, then sometimes the cells you have a doubt. Maybe because of temperature they may not grow. So everything you should follow the same pattern except the plasmid addition. Remaining everything you should follow. Added 1 ml. 5 minutes over.
5 minutes more.
Okay, fine. You just add 1 ml of LB. You label one as a negative control, no?
C is a negative and P is a negative.
Okay, fine.
The competent cells with the vector, they are in the plastic, and we have kept them in the water bath. So the 44 degrees is enough to
Penetrate.
like enough.
Yeah, it's well
Because the plastic is also there.
plastic.
Plastic, no, the ependoptube.
No. Anyway, cells are present inside the ependoph only. We are not blocking in
No,
between.
no, I mean the heat is enough. So that
Yeah,
it
it is
passes through
enough.
the plastic.
It's well fine. 45 to 60 is more than enough.
He's saying that we are using plastic tube.
Yeah. I can understand.
There is
Enough.
Is there any chances of plastic leakage to the
So he's telling temperature is enough to transform or not.
Because there is a barrier also.
Yeah. Still it's enough. We tried, it worked. So if you, in your case, if you try, if it doesn't work, then you should try it for higher time. But if you're going for higher time, it will kill the cells. They are very sensitive to temperature. So 45 to 60 is more than enough. I'm giving 30 seconds extra.
Our plasmid.
Yeah, add the respective cells to the respective tubes. That is very important.
Next step.
You
You
trans-
broth, no? I'll be broth. Incubate 1 hour, 37 degrees with constant.
No, she should transfer that cell to the this. Yeah, negative control to the negative control tube.
that only competent cells and vector is added anything?
No, you can use 1 ml tube. Entire sample you transfer. What do? You take 1 ml, transfer like little to the competent cells. Okay, entire thing, and let's take treatment. You are going to start. Take little media from here. Don't touch anyone. It will contaminate. Take little. Okay.
Competent cells
Keep it open only.
integrate.
Keep it here. Which sample? No, sample. This is negative control.
E. coli. Okay.
You don't confuse.
Not to the integration.
Mix it
Understood.
and take the entire thing. Slowly. Gentle. Be gentle. Take entire thing slowly.
What is added?
We added 1 ml of LB media. Mixing and transfer to the bigger tubes. It should grow in them for the recovery. Slow. Treatment, right? This is treatment. Okay, now similarly you change the tips. Yeah. Don't use the same tip. You will get contamination. Now you do the same thing. You take some ml of media. No. Tip. Huh?
Sterile medium.
Yeah, sterile media.
Competent cells and the vector.
Hmm.
And negative control is
Without plasmid.
without plasma.
Same competent cells without plasmid.
And positive control.
Positive control, if you have any other plasmid, you can use it. At least negative control is very important to check whether contaminations are there. Sometimes ampicillin, as I mentioned, they are weak antibiotic.
And after this, we will do plating
Yeah. Finally, we will be plating it,
on ampicillin.
ampicillin plate, and we'll keep it overnight. Tomorrow morning, you can see the colonies. 1 day it takes to form the
In
colony.
this colonies, what we do?
We'll go
Yes.
again. We'll check the colony PCR to find out. That we are not doing, but I'm telling it. You should do it to confirm. For now, we are going to. I'm just teaching you the transformation into E. coli. That one step, and another one transformation into the algal cells. These two transformation I'm teaching you.
In that large tube, there is LB bot.
Transferred, right?
Yes.
Now we need to grow them in 37 degrees Celsius for how many?
37 degrees.
1 hour.
hour at 37 degrees.
Shreya,
Constant.
need to keep in Narendra's lab these two. Keep these two in the incubator. 37 degrees Celsius. You just say E. coli.
Static condition.
It's shaking.
Condition.
But it's occupied, no?
I don't think so. It should be.
Fine. You can keep it there. 37. It is 37 or what? If you want to remove, you can keep it here. Then set 37 and keep it here.
The temperature maintenance will be done for this
Which one?
incubator shaking.
Yeah, 37 is very important.
And on the next day, we have to isolate the plasmid.
From these plates. From these plates. We need to pick a single colony, inoculate in the LB media, overnight again. From the LB 10 ml media, we will isolate the plasmids and again go for algal transformation.
We
Okay?
will transfer the plasmid to the algae.
Yeah.
So now you have already isolated.
Isolated plasmid, so we are directly going. So I am bypassing the steps. That's it. Any doubt in the steps?
So from this gene of
నువ్వు
interest,
చేసేటప్పుడు చిన్న ట్యూబ్స్ లో ఉంది కదా
Sorry.
what you are doing here?
దాన్ని ఫస్ట్ కొంచెం ఎల్బీ మీడియా వేసి
Right now, that's what, sir. We are transferring in the plasmid. For example, your gene of interest has been cloned into the vector. So now you want to multiply those copies, right? One or two plasmids only you have, but you want to multiply them into hundreds, 200s, more concentration. For that, you need to transform into E. coli. Because E. coli doubles faster, makes multiple copies, right? So that's why we are transferring into E. coli.
what we will do
That's step one.
Now this algal culture, what we are doing in algal culture, gene of interest.
Yeah, finally, the entire aim of this program in this molecular biology is to transform your gene of interest into the algae. For that, we are doing all these steps right from RNA isolation. We will be isolating what gene you want to amplify.
Okay.
You need to amplify the gene that we did on that day. After amplifying the gene, you need to clone into a vector. That we are doing. We should have. I explained you theoretically how it has to be done. Then we need to isolate, bulk it up, that cloned plasmid, and finally we need to transform into the algae. It's all a single sequential experiment has to be done. It's up to you. It's not necessary that it should be very fast, but you can complete it if you work hard.
How much time it will take?
It will take a week.
ఆ
That's it.
తర్వాత 1 ml మొత్తం పెద్ద దాంట్లో ఆల్రెడీ చిన్న వాటిలో
Yeah, yeah. Algal, and after transformation into algae also, it will take week to grow. Algae grows slower. From that also you need to do screening, multiple screening.
ఆ తర్వాత పెద్ద దాంట్లో ట్రాన్స్ఫర్
పెద్ద దాంట్లోకి ఎందుకు ట్రాన్స్ఫర్ గ్రో అవ్వాలి కదా
Yeah.
చిన్న దాంట్లో వేసినా కూడా
You're doing chloroplast.
Bro,
చిన్న దాంట్లో వేస్తే
nah,
grow
Vijay,
ఉండకపోవచ్చు.
we can go for break now.
And the pen-
Arrange it for we have 1 hour break.
centrifuge
1 hour, 1 hour time.
both are centrifuge tubes, right? Because
We are doing chloroplast isolation transformation today.
Yeah, the final after this plating, I will be showing you how to do the transformation using electroporator. Tomorrow, Malbio ends today.
Okay.
Tomorrow something. That my lab members will help you.
Instead of Thylacard, we have isolated.
Tomorrow we'll run. Tomorrow we'll do, sir. Yeah, tomorrow we'll do. So yesterday, how was it? I left in between after Thylacard. How did you see the peaks?
Yeah, very nice. They have
Ah.
just
we got very nice terms from
Ah.
PS2 and PS1
That's 77.
You got 2 peaks. Jyoti Bhaiya, they got a second peak also. Better than us. Lower. We did it or yesterday what? That means because of the higher concentration. You guys are so lucky. Because I did it like last week. I didn't got that good.
7 micrograms.
You have a photo? Jyoti Bhai, you have a photo? Okay. So you plotted also, you guys plotted, sir.
Not plotted, but
today they will show how to
Oh, you are very late yesterday. When I was leaving, it was 6 something.
20 minutes.
But CD is a very good technique. And with liquid nitrogen particularly, you should be very careful.
not serious.
77, sorry. 77. I was confused. I think almost our experiments are near. BN tomorrow, BN. After BN, tomorrow it will be completely BN. It takes a long time for running. And after BN, I think HPLC, one more experiment is there.
ఇప్పుడు ఈ step
CD is not there. BN is there.
shaking consistent shaking ఇప్పుడు షేకింగ్ ఇంటిగ్రేట్ మళ్ళీ తీసి మళ్ళీ 1 hour సెంటిగ్రేడ్ ఆ తర్వాత ఏమంటే అప్పుడు రాస్తాం
Nice. I think Jyoti Bhai, we need to increase our concentration. I think
ఇప్పుడే
5 is not working. Next time we'll go for 10. Yes,
One competence will happen.
but should not create that much difference. So what you understood? Why yesterday everything you understood about 77K? What it is done? Why it is done?
PS1, PS2.
PS1 and PS2. PS1 particularly. We can't see the PS1 bands in the normal.
That cryo
At low temperature, very low temperature, you can see that.
using the quartz.
Yeah,
Cuvette in the sense that is an enamel tube.
in high temperature it can
very quad zone.
Quartz is an enamel tube.
Mm.
It is
Oh,
nuclear magnetic resonance.
yeah, yeah.
pepper
Enamel, you biologists might not be knowing. no?
Yeah, I haven't used it. That's
That's
right.
why we are seeing the biology as well as
Yeah.
రాసుకున్నాం అంతా
But the enamel, that is enamel they are using actually for spinning.
Okay,
They
okay.
are enamel, we have to our synthetic in the laboratory they are synthesizing the compounds.
Okay.
So synthesizing compounds, they will see that they will remove some groups are there. They will
Okay.
then they put and they see the enamel.
Oh.
Scaffold, what is the enamel for the enamel? And then initials, then they can see the peak. The magnetic resonance, nuclear magnetic resonance.
I'm not that good in that side. NMR. Yeah.
This is also there are two parallel things.
Yeah.
That is chemical characterization.
Yeah.
And biological characterization through protein.
So morning class, you guys understood something?
Yes.
Chloroplast transformation, they understood something.
Yeah, you are nice.
Oh. It was like a last minute preparation. I was given a last minute. So I was in a hurry to make.
That much nice you are
Well,
presenting.
And the topic is very small. It's not a big topic. Very narrow down topic.
These are very good things for you. That,
I used to do. My seniors are there. Few are right now not there. I am just doing CRISPR.
1000 rpm time 1 hour
Interesting.
I made that side because
Since then,
It's fine. Okay. What is the next step? Fine, very good.
Weight also should be.
Pre-suspense cells with 100 microliter of LB media and spread it on LB plate with another syringe.
Yeah.
100 micrograms.
LB plates are here. Okay, but it again I'm telling this has to be done in the lamina. For our convenience, we are doing it outside. It's a E. coli. Definitely, it should be done in lamina. But for now, we are doing it outside. Okay.
No, we don't have laminar.
We'll just you have a
Here
spreader.
we don't have laminar.
Spreader.
Even in the lab is not there.
That is not working. We will go down. We have culture room. That is there. And in the laminar also, these many people should not stand. There is no point. So almost we will spread it and we'll keep it in the 37 degrees Celsius, right? Overnight. And this step is done here, and then we'll go for algal transformation. We need to little bit speed because that electroporation we need to go to other lab. Before they leave, we need to. Otherwise, tomorrow. I'll show you once again. Not at all charged.
Move like this now. It will come noise. Or
Okay, let me brief you about the transformation, algal transformation. Just can you see the screen there? Oh, it's not moving.
maybe different time away.
And okay, yeah, now we can. So electroporation, how many of you know the principle of electroporation? Morning I was talking you about two transformation method, right?
No, but plus or minus voltage, some particular voltage that cells pores will be open for that.
That's what I said. Morning I haven't talked about the
So that
electroporation.
pores the GN means the vector will be.
Yeah. So why we are using here, now I am focusing on the not chloroplast transformation, nuclear transformation I'm doing. For nuclear transformation, you can use electroporation. So here, what is the principle? As she was telling, we will be giving a short high voltage pulse, electric pulse, that is going to break the cell wall. So by this, we are creating a permeable pores, and at this time, whatever the extra chromosomal DNA, in this case plasmid, will be allowed to get inside the cell. And once they are inside, how the recombination happens in terms of nuclear? Is it homologous or non-homologous? Random means non-homologous. Be prepared for this kind of questions. So non-homologous question. So non-homologous recombination will happen. So randomly, they will just incorporate it into the position. So then you need to screen using a PCR where they have been inserted. For example, you want to interest in a particular gene, then you need to amplify the gene near to the gene. By seeing the shift in the bands, you can confirm, okay, out of the 100 colonies, 5-10 colonies have a insertion at the particular position. You need to screen colonies which are very specific to the insertion. That's your target, right? So that's the image you can see clearly. When electric pulse are given, they used to break open the cell. As a result, based on the inverse polarity, they used to enter whatever the genetic material they will enter, and then whatever mechanism you can even send Cas9. If you are given Cas9, they will tends to break the cell. Double stranded breakage will happen, and if you are not going to give any Cas9, then random incorporation will happen. It's up to you whether your interest is just a overexpression, then Cas9 is not required. But your interest was to create a mutation, then you need to send the Cas9. Fine. This is the protocol. So this will be the instrument electroporator, and this is the main instrument, and this is the chamber where you will keep your cuvette. Okay. And this is the processing. We will be taking some 100 ml of cell roughly, CC1, like a well grown, around 0.6 OD, and we'll be pulling down, harvesting, and that cell we'll be resuspending in a buffer, max efficiency transformation reagent. That's a commercially available reagent that used to
Kit only it will come.
no, it's separately you can get it. This is a reagent. The main purpose is to remove the cell wall.
What is the maximum efficiency reagent? What is the composition of that reagent?
That is commercially available, sir. I'm not sure exactly about.
Sometimes they should mention.
No, you can check it. It's an in vitro gen product. Majorly they use it for the cell wall removal. Whatever they may have enzymes to remove the cell wall or
I see,
something.
sir.
You can check it. In vitro gen max efficiency reagent. You can type it. You will get it. Okay, so you will be washing that with twice with the max efficiency reagent, followed by you will be incubating your plasmid. In our case, it's a PSL18, right? We will be incubating them, and then we'll go for the pulse in the cuvette, which we have a specific cuvette, and we will be giving a pulse that is 300 volts, very high pulse. 2 times we'll give, and this pulse varies to different methods. So this is like 300 is generalized protocol, and there are multiple. If there is a different instrument, different pulse. Cannot set the program for 2 pulse simultaneously. We can set only 1 pulse. So according to this protocol, I have given 2 pulse 300 volt for 8 milliseconds. So milliseconds means you can measure how short span. 8 milliseconds you are going to give 2 pulse, and immediately after that you need to transform them to a tap media, and it has to be kept in dim light overnight, and the next day it has to be plated in the tap plates. Okay, this is the protocol we will be doing immediately after this. Okay, is nothing but tris acetate phosphate. Generally we grow the Chlamydomonas in the tap media photoheterotrophically. We supply the acetate, tris acetate phosphate. And finally we will be confirming the mutants. This is a plate which I generated. This is again negative control, positive control, and this is the actual plate. In terms of positive control, we can see colonies, whereas negative control there is no colony. This is a transformed cells, and we will be confirming using either PCR, RT-PCR, and localize. If your vector has any fluorescent tags, you can visualize them in the confocal microscope. Okay, fine. Let's stick to the E. coli transformation. Now what's the step we need to do? You need to remove the media, right? Yeah. Who is going to do that? Remove the media and add 100 microliter of fresh LB media. You want to do?
Need to
What is the step?
resuspend with 100 microliter of
Yeah. Use this as a. That's what you remove. Use a pipette, 1 ml pipette. Use a 1 ml pipette. Remove the entire media. Only the cells will be at the bottom. No, without disturbing the cells, you remove only the media. Use 1 ml.
discard, sir.
Yeah. Only supernatant. Only the media. Don't disturb the cells. Remove entire media, complete media. Fine. Now you another
A bit is there.
fine. Otherwise, you will disturb the E. coli cells. Change the tips here. Can see. She is good in handling the pipettes. See? She should be very slow in releasing the pipettes. Should not be harsh. So now you add 100 microliter. Maybe you can use the different. Use this. Yeah, 100. No, that's it only. Just pull it, push it in. No, here on. Here only. What happened?
He thought it was chloro.
I thought
I didn't
chloroplast
micro-
It was written.
Oh, no. This is nuclear transformation. Only difference is both are similar. Only difference, the transformation method, there we use ballistic. Here we use the electroporation.
And we media 100
Yeah.
I see. The schedule of chloro.
Should I
This yeah, completely solubilize it.
I thought chloroplast.
Schedule it is chloro.
Okay, maybe initially, but almost similar. Nothing. Because of the instrument, we will go for this. But apart from this, you don't feel any difference between them. Only the instrument, how you are transferring them, that's a major difference. Yeah, fine. You go for the other one.
Ballistic one, here it is.
We don't have. We don't do actually chloroplast transformation in this university. If you want to do, we can look for ballistic and PEG mediated. We can do PEG mediated. Depends again, if you really want to do it, you can do it. No, it's a growth media.
Chloroplast in this lab regularly we used to do.
No, not regularly. Whenever we have some, it's not regular. No. Once we generate, then we'll. Chloroplast transformation we haven't done. Nuclear we do. Nuclear transformation.
They are working on photographs.
Yeah, but it's not necessary that only chloroplast genomes is located in the nucleus also. That part also we work. Because already chloroplast related genome already well characterized.
Place where
Yeah, it's a growth media where you grow, no? Yeah, sucrose is just what can I say? Extra carbon source. Understand? Then who want to split it? You did it already, so don't come again. Let's go give a chance for other guys. Any of you want to plate? You want to plate? Huh? So this is the plate, and again I'm telling you have to do it in the laminar. So take 100 microliter. So in case if you are taking treatment, you mention control or treatment, whatever sample you are taking, and use the pipette 100 microliter. First you aliquot into the plate. above it. Then use the spreader and spread it. That's it. Okay. But you carefully note down which sample you are taking. Is it a negative control or positive control? You need to label that. Which one? Yeah.
Just remove all the media.
Tomorrow you can see it.
Tomorrow you can see.
Same media. 100 micrometer.
Fresh media. Yeah.
Don't speak much and you be fast when you are opening. No. Particularly after opening, you don't keep it open for a long time as we are outside. So be fast. First you remove the plates. That's very important. Okay. And this also
Antibiotic added.
antibiotic added.
Out.
Sorry. Because you are telling when you will add that. No. You should not expose this much. You should wait. I'll tell you. What I will tell. Give me. Anyway, I'm doing it outside, but still. This is negative control. Okay. I'm taking negative control. Okay. Even if you are doing it in the laminar, open this much only. That's it.
So that's also negative only, right?
Yeah, this is negative.
I mean,
We need to label it. I can label it later. Wait. Now you cannot invert. Okay. Fine. That's it. You can
Once
discard.
they've added 1
100 microliter.
then the spread.
Yeah. Now you label this. This is negative control, right? Okay. Yeah. Mm. You have one more plate or that plate? Show me. Media is very less. This is better. Use this plate. The way I added, similarly you add it. I will give you the spreader once again. Use this. No, touch it down. Very gentle while taking it up. Close it. Discard that. What happened? It was not there. Okay, leave it. Keep it. Oh, it was there. Fell down or? Okay, fine. It's not. Yeah, spread it. Spread it like how I did it inside. Don't give too much pressure. Yeah, fine. Fine. Throw it there. Yeah.
this one is
We need parafilm. You need
label it.
sample. Make it a sample. Where is the? So I need a parafilm. 2 parafilm. Here. Scissor. Here also we have. Cut it fine like this. This size. This size. Wait. This size. Whatever. No, fine. No, not this much big, sir. Fine. That is enough. Yeah. More than enough. Yeah, someone want to grab it?
Fine, it's okay. It's showing.
He won't show. He will only do. Just invert it.
Stretch it.
Stretch it.
Then from the beginning, from
Yeah.
starting,
Stretch it. Be fast.
Don't give over.
Yeah. Same. Keep it from there. Slow. Need to join that. case LB sample. Okay, still it's not sealed properly. Yeah, that. Okay,
It's open, sir. Take. Sir, take.
give me,
Here, it is open.
give me.
Salty.
It's fine, let's work. What? She's afraid that it will spoil the sample or something. Okay, now it has to be kept in 37 degrees Celsius overnight.
You're keeping?
So yeah, 37 degrees Celsius. That's it. Okay, now we'll go for algal transformation. Okay, so I have already collected the cells. 20 ml of cells I have collected because this is OD, it crossed above 6.6. So the cells are already more. So I have taken less volume cells, 20 ml.
What OD? The 0.8?
0.6 is the ideal. 0.4 to 0.6 should be the ideal OD. And if it is 0.4, then you collect more cells, 100 ml cells you need to collect. Whereas this is above 1, OD above 1. So I reduced to 20 ml. But I suggest you, because we didn't specifically keep the culture for transformation. That's the reason we are using the old cells. But when you are going for actual transformation, I suggest you to keep a fresh cell and always stick to the OD of 0.4 to 0.5. Keep 100 ml cells, stick to 0.4 to 0.5. That will be the best OD where the cells will be in the mid log phase. If it is like old cells, then probability of dead cells will be more. So I'm telling you before itself, when you are planning for actual experiment, try to be mid log phase. So 1 day later. So yesterday it should be 0.6, but it was today. So 1 day old. So but when you plan, exactly Chlamydomonas 0.6, 0.4 to 0.5 will come in second day, end of the second day. So something around, I can say 35 hours something, it will come. So you plan your experiment in a way that you will start the transformation at 0.4 to 0.5 OD. So now let's go for the algal transformation protocol. I think we need to harvest, wash. No, I elected this one only. So first we will be harvesting. We are going to harvest this. Wash twice with the tap max transformation efficiency, right? Okay. We need to harvest them. Come, I'll show you how to do the harvesting. Okay, Raghendra is there. Help Raghendra. Put it for centrifugation. You know how to use the centrifugation, no? Set it low temp, like low speed, low RPM, around 3000 RPM, 5 minutes.
Sound, right? Oh, here only.